第二个 section 呢是由我们国外意大利的讲者 Dr Davide Manis 来为我们演讲。那他的题目是 Tackling COVID-19 through Syndromic Testing, QIAstat, Diagnosis, Respiratory SARS CoV2 Panel 这个题目很长，你就知道这待会呢一定是很精彩的演讲。那我先介绍一下May I now introduce Dr Davide Manis to our audience. Dr Davide Manis is a medical doctor with a unique mix of the expertise combining clinical and public health experience with medicine and clinical affairs leadership roles in both pharma and diagnostic sectors. His key expertise focusing on aligning diagnostic drug development with clinical and public health needs. So now he is currently the Vice President, Head of Medical Affairs and Chief Medical Officer, Infectious Disease and Immune Diagnostics at QIAGEN, and now he's leading the global medical affairs functions across all the community to represent the company to guide front-loading for development of novel assays. So now let us give a very big welcome to Dr. Davide Manis for the topic. Okay, Davide, we will now pass the time to you.
Thank you very much, Dr. Wang, for the very kind introduction, and I'm delighted to have this opportunity to present today. There is a recording, so obviously the introduction to my presentation is summarized there. What I suggest is that obviously I can take all the questions at the end of the presentation. The presentation will focus in providing a background on how we came to develop a respiratory panel inclusive of a SARS CoV2 COVID-19 solution. And it will also elaborate on how we position syndromic diagnostic and the potential role of syndromic diagnostic within the COVID-19 pandemic. So I suggest perhaps now we get into the presentation and into the recording for the sake of time, and I'll be delighted to take questions at the end of it. Thank you very much, Dr. Wang.
所以待会我们会先看录影带Dr. Davide 预录的，那各位看完以后有什么问题就直接在我们的网络上呈现，他待会也非常乐意回答我们问题。Okay, now we'll play the video.
Thank you. Thank you very much for the invitation. As mentioned, I'll be touching on syndromic testing within the context of the COVID-19 pandemic. While I will be focusing on syndromic testing, I will spend the first 10-15 minutes of the presentation really to focus on testing and how testing has evolved within the context of this pandemic. I will touch on 4 topics, and as I mentioned, I really like to highlight the diagnostic paradigm and the peculiarities around SARS-CoV-2, and particularly COVID-19 as a disease, which have placed us in front of a challenge, particularly presenting very acutely at the beginning of the pandemic, and how we have reacted to this challenge, both as QIAGEN as a company, but particularly with the healthcare system and manufacturing around the world in addressing the acute PCR need. Now, while this is a novel pandemic caused by a previously unknown virus, COVID-19 does resemble a familiar phase. As you're aware, SARS-CoV-2 is a beta coronavirus and is related to other coronavirus, which we've become familiar with over the last decade, SARS-CoV-1, MERS-CoV, and other common human coronaviruses, better known as seasonal coronaviruses. As all coronaviruses, it has a single-stranded positive stranded RNA genome, which can be split into genes which encode for non-structural or structural proteins. In common with other RNA viruses, as we are now well aware, is the frequency of erroneous or ineffective replication of the genome, which can lead to potential mutation. Fortunately, to date, despite the noise around it and despite a very close monitoring of the variability, no mutation or group of mutation have seemingly led to altered properties or phenotypically relevant variants. There are, however, key differences between SARS-CoV-2 and other respiratory viruses for which we have become used to and for which we have in the past prepared from a pandemic perspective. Now, like all seasonal coronaviruses, as well as SARS-CoV-1 and MERS, the SARS-CoV-2 is part of the coronaviridae family, and the majority of these coronaviruses of human relevance, particularly the SARS-CoV-1, as well as the seasonal coronaviruses, were part of the syndromic panel that was available in the market prior to the pandemic on the QIAstat platform. From a clinical presentation point of view, while many symptoms overlap among the respiratory viruses that are depicted here, there are certain peculiarities around SARS-CoV-2, and particularly around coronaviruses in general, which compared to flu and RSV all have a longer incubation period of up to 2 weeks, which really has made COVID-19 more complicated from a control perspective, in addition from that paucisymptomatic or pre-symptomatic phase where infectiousness and transmission of disease seems to be occurring compared to other respiratory infection. When it comes to case fatality, it has been very well established for flu and RSV, with, of course, with seasonal variation, but with an average of below 1% case fatality for flu. Now, when it comes to SARS-CoV-2, case fatality figures are really still a matter of discussion, given the difficulties in establishing a reliable denominator. And I think this really highlights the difficulty in diagnostics we had at the beginning, we're still encountering on a global level, where there is this disparity in assessing both case fatalities and incidence, given the variability in denominator linked to the testing capacity within different countries and markets. Another key unknown, which I would like to highlight towards the end of the presentation around not coronaviruses in general, but particularly around SARS-CoV-2, it's the issue of seasonality and the potential for SARS-CoV-2, COVID-19 to develop into a seasonal respiratory infection. But we'll touch that later in the presentation. Now, while a detailed discussion on the clinical presentation of COVID-19 goes beyond the purpose of this presentation, I think it would be of interest to highlight some key peculiarities around the natural history of the disease as we are learning. Again, I think this is a learning curve for all of us, from microbiologists to clinicians, to manufacturers themselves in understanding how the disease is evolving. Now, what is becoming clear, and I tried to oversimplify with this figure, is that COVID-19 presents as two overlapping and consequential pathophysiological events. These two events are, as you see on the left in blue, a very typical early viral response with high viral loads, and a symptomatology which I would dare saying it's very much overlapping with influenza-like illness, presentation with fever and cough, and mainly upper respiratory tract infection symptoms, and sometimes overlapping with lower respiratory tract symptoms. However, in a portion of patients, this evolves around day 7 to 11 into a more inflammatory response and a dysregulated immune response that has become the dramatic clinical presentation of the very severe cases of COVID-19, presenting as acute respiratory distress syndrome, ARDS, or systemic inflammatory response syndrome, SIRS, which are being commonly called and identified with that so-called cytokine storm, which has really put a tremendous burden on the healthcare system, particularly in that portion of patients which dramatically progress toward this exaggerated and dysregulated inflammatory response, with the difficulty from a clinical management point of view, particularly in terms of establishing mechanical ventilation and trying to mitigate the dysregulated immune response. Now, while this presentation from a clinical standpoint has no diagnostic implication as of today, because we do lack any validated prognostic test, it does have implication on the choice of type and timing of treatment, ranging from viral suppressive treatment, particularly in the early phase, to immune suppression and modulation, which is required particularly in that phase of dysregulated immune response and exaggerated inflammatory response. Obviously, prognostic and profiling from a diagnostic standpoint would be really helpful in identifying very early those patients that are entering this dysregulated immune response phase and would therefore benefit from immune suppression or immune regulation. But let's focus for a second on the actual individual diagnostic positioning of testing for COVID-19 or identifying SARS-CoV-2. What you can see in this illustration is clearly a depiction of the core and pivotal role that PCR plays and has been playing. It's been playing a key role in early identification of cases and has been the cornerstone of the entire diagnostic response to the epidemic. With the stress on laboratory and manufacturing capacity that we all become familiar with, obviously overlapping with that PCR role and kinetics is antigen detection. I think we are still behind in catching up with the development of sensitive enough antigenic testing, but this is progressing fast, and we hope that soon antigenic detection alternative to PCR will be available, with all the advantages that you are probably familiar with antigenic platform being friendlier from a ease of use point of view, turnaround time, and ability to be in proximity of patient or even to be used in the community. The other very controversial role from a diagnostic, particularly from a clinical diagnostic point of view, is actually serology. Now, serology has obviously played a role from a surveillance standpoint in assessing the percentage of population, really from an epidemiological point of view, that is exposed or has been exposed to the disease, but has yet to be positioned from a clinical diagnostic point of view. Now, when it comes to the different antigenic response, again, I would say the jury is still out in really understanding the specific of the kinetics of the antibody response. But what is really becoming clear from a peculiarity point of view is that in a dissimilar way to what happens with other respiratory infection, what we are used to in seeing of early IgM response identifying acute phase of disease is not so visible with COVID-19, with the IgM and the IgG detection being almost concomitant or differing by 1 to 3 days, and therefore we are lacking that window of acute phase identification that we're used to with other respiratory infection. And lastly, let me just also position diagnostic from a more control perspective, and really looking from this point onwards, what will be the diagnostic need in terms of testing for COVID-19 and identifying a SARS-CoV-2. This slide is really the outcome of an internal brainstorming, which I'm very keen to share with you. Our feeling is that from a control perspective, the stress on the need for PCR and potentially antigenic testing, and eventually serology, if that is better positioned from a clinical diagnostic point of view, can really be in a way forecasted by two key community drivers. Firstly, to the left, just a simple graph to depict the situation that most countries, with a few exceptions that are still facing a peak of the epidemic, are now facing, which is the release of and the lifting of the COVID-19 containment measure, and really the need to rapidly identify isolated outbreaks. And that will be probably the driver in terms of diagnostic need from a community perspective in the months to come as we enter the next autumn and winter season, particularly in the Northern Hemisphere. And that's probably where we can sort of identify the second driver for the diagnostic need as the epidemic evolves, and that is the potential need to have a syndromic approach, or at least a multiplex approach, to be able to identify as we come into the flu season, and there will be an overlapping of several respiratory infection, the ability to quickly discern within the ILI, that is influenza-like illness presenting in the emergency department or in primary care, to be able to discern between flu, RSV, COVID-19, and several others we will see later in the presentation, respiratory infection, and therefore the ability to implement infection control, but as well, in selected cases, treatment interventions. Let me just move now on the specific, on this note, on the syndromic need, on the specific of the syndromic platform that is namely QIAstat DX, that will be matter of discussion of the next part of the presentation. Now, before entering the specific of our syndromic platform, I would like to highlight that the response to COVID-19 at QIAGEN goes beyond a syndromic solution. QIAGEN supports several testing protocols by providing test components that includes mid to high throughput automation solution, and obviously the multiplex syndromic detection solution represented by QIAstat. As you're aware, QIAGEN, and it is listed here in a succinct way, QIAGEN portfolio of RNA extraction solution includes kits that can be automated on the QIAsymphony platform, as well as the EZ1 instrument and QIAcube. And for those of you who have been involved in the pandemic very early, you could have seen that extraction kits were and are still part of several protocols for LDTs or reference laboratory protocol for SARS-CoV-2 identification. Obviously, as a company, from the very beginning, we had dramatically scaled up production to really respond to this emergency. We moved to 24 hours, 7-day-a-week operation, and are investing in additional capacity, particularly in view of the upcoming winter flu season, which I mentioned could see a dramatic increase in the need to discern presenting ILI to both primary and emergency care. Now, from a syndromic perspective, there have been a rapid engagement from QIAGEN point of view to deliver on what became really the first to market complete syndromic solution, integrating SARS-CoV-2 to what was already in the market, with either 20 under the FDA approval or 21 under the CE marking respiratory pathogen panel. Now, prior to, as you might be aware, prior to the inclusion of SARS-CoV-2, we had two products in the market, as I had mentioned. One, a CIBD mark product, which I mentioned with originally 21 targets, and one FDA with 20 targets. We will go over the targets in the panel later in the presentation, but just for just placing the advent of the complete SARS-CoV-2 respiratory pathogen solution, two key dates are depicted here. The CIVD marking on March 18th, which was followed with the support of a U.S. government grant through BARDA, was followed by the FDA Emergency Use Authorization, EUA, on March 31st. For those of you who are not familiar, I just want to give you just a pictorial overview of what our QIAstat diagnostic analyzer looks like. You can see there are two main components: an operational module, which presents a very intuitive module with a simple graphical user interface, and an analytical module which supports real-time PCR technology with the cartridge entry points. And of course, that analytical module can be expanded to 4 modules, can be customized according to the throughput needs to be able to process 4 samples at the same time. Now, at the core of the QIAstat instrument and solution is really the cartridge. The cartridge contains all that is necessary to process the sample. There is no mixing of buffer required, simply loading of the sample, which can be in liquid format or direct swab into the swab portal or the liquid portal with minimal hands-on time. The hands-on time in preparing the cartridge is calculated at less than a minute, really. As I mentioned, this is a multiplex solution, so the ability to detect up to 48 targets is really at the key of presenting QIAstat as a true syndromic solution with a turnaround time of approximately 1 hour, 67 minutes for the respiratory pathogens, therefore really presenting as a rapid syndromic diagnostic solution, which can be positioned within the context of a pandemic in a very advantageous way in providing a rapid turnaround time for differential diagnosis. As I mentioned, the ease of use is also at the core of the positioning of QIAstat as a frontline solution. You can see here in this flow the ease of use. Here we have the version with the liquid format, usually with an aliquot of around 300 microliter of UTM, universal transport medium, which is inserted in the port. The cartridge and the sample are both scanned through the QPR code, and the cartridge is then inserted in the cartridge port, and that's as much hands-on time that is required to then enter in the processing of the sample for 67 minutes to results presented by the user-friendly interface that I've shown you earlier. Now, one key characteristic of QIAstat is the ability to provide CT value. But before going into that, what I would like to remark is that QIAstat provides qualitative results, and that is one of the feature is revealing the amplification curves and the CT value. But the regulatory indicated use remains a qualitative one and not a quantitative one. However, and I always like to quote the words of one of our early users of QIAstat, the ability to provide both the amplification curve as well as CT value, particularly to the laboratorium microbiologists and molecular biologists, it's almost like removing the black box around the more traditional syndromic approach and giving more information on every sample tested. As you are aware, CT value and amplification curve can provide rationale and explanation when complex or unexpected results are received, and as well providing input when a contamination is suspected. We will come back to the potential role of CT value into the COVID-19 epidemic, but it's definitely something that is gaining a lot of attention from a patient management point of view as well. Let me now transition in really the development of the SARS CoV2 panel. When QIAstat respiratory panel was initially designed, all the primers and probes for the respiratory pathogen were contained in reaction chamber 1 to 7. You can see here at the bottom of the cartridge, there are 8 reaction chambers. So in the respiratory panel that was on the market, we had 8 channels, of which only 7 were occupied. And therefore, the 8th chamber was left empty. This proved to be beneficial to us during the outbreaks because we were able to add the primers and the probes for the 2 SARS-CoV-2 target in the 8th chamber by themselves without interfering with existing targets distributed in the other 7 chambers. So this gave us an advantage in terms of time to development, as we knew we were not going to interfere with any of the other targets, and really in the ability to bring very rapidly the product to the market. I mentioned the product that was already in the market with 20 or 21, depending the region of approval. You have here a very comprehensive list of those 19 viral and 3 bacterial respiratory pathogen, which are really encompassing probably the majority of the culprits for influenza-like illness and acute presentation of upper respiratory infection symptomatology to the traditional ER department, or I would say even in primary care. Now, when it comes to the SARS-CoV-2 targets, on this slide, I just want to show you a schematic view of the SARS-CoV-2 genome to help explain how we selected the SARS-CoV-2 QIAstat assay targets and how these compare to other design. The proprietary QIAstat SARS-CoV-2 assay target the ORF1b, also known as the RNA-dependent RNA polymerase region, RDRP, and the E gene, which is very similar to the WHO, but different than the US CDC, which targeted only the N gene, and other publicly available designs which were implemented very early in the epidemic. Now, the basis for us choosing the E gene and the RDRP genes was the alignment with available sequences at the time, back in February, and the fact that the combination of these two targets really maximize sensitivity without creating any cross-reactivity and therefore jeopardizing specificity. So it was really within the context of our cartridge approach and multiplex approach. It was the combination that achieved the maximum sensitivity, which was in analytical term achieved at 500 copies per mil as a limit of detection. We also went further in assessing really the alignment of our primers to the variants that are arising. Particularly, this is an exercise that we do on a weekly basis in aligning our target primer sequences to the variants that are being uploaded on publicly available database. This is a report from a paper that we just published on the International Journal of Infectious Disease, where we assess the sequence alignment as of week 21. But again, we are continuing to do this. And as you can see here, as of week 21, we assess both our panel design as well as 4 public available primer sequences from WHO, CDC US, China CDC, and the Hong Kong panel, and we assess them against the available sequences as of week 21. As an outcome, what evinces here is that we, out of a total of just over 33,000 genomes, we 99.1 for the QIAstat SARS-CoV-2 sequence matched with 100% coverage of all oligonucleotides. Of the 301 genomes that were not aligned, 35, that is 0.1%, contain a variation in a critical position of any primer or proband, which brought us to assess the potential impact on sensitivity in vitro, and it was very clear that our limit of detection was not affected for this 35 critical variation. As a result and as a conclusion, and you can refer to the paper, what we demonstrated here is that the combination approach which we chose of combining the ORF1b and the E gene was the combination of more than one assay targeting real-time reverse transcripted PCR for SARS-CoV-2 panel can mitigate the risk of loss of sensitivity and potentially specificity during this outbreak. We're continuing to monitor, and as of today, we haven't identified any key variants that would impact on the overall sensitivity of the assay. Very briefly, what I want to highlight is three sets of data that have been either utilized for regulatory purposes or through independent study. This was a limited set of data that was utilized for the initial CE marking with a study carried out in the Bichat Hospital in Paris, France, where we demonstrated, albeit with a small numbers here, PPA and MPA percentage positive agreement and negative positive agreement, negative percentage agreement, sorry, of 100%. The study was then replicated independently as well by Bichat Hospital, where the sensitivity was replicated over 40 samples with a sensitivity overall of 100%. And a specificity of 93%, again, in an independent study carried out by the same Bichat Hospital. And lastly, the data utilized for the EUA approval by the FDA, where the analytical sensitivity was replicated and confirmed at 500 copies per mil. Analytical specificity again was confirmed with no cross-reaction in silico versus any of the other human pathogen, or most importantly against the seasonal coronaviruses. The clinical validation was performed as per FDA guidance on contrived clinical sample and confirmed what we found in the CE marking early data, replicating a PPA of 100% as well as a negative percentage agreement of the same value. But now, having provided this quick overview of the QIAstat syndromic solution and how we came to quickly combine the SARS-CoV-2 target with the existing 20 or 21 targets, let me just now enter the final portion of my presentation, where I really want to spend some time on the potential value of syndromic testing within the COVID-19 pandemic. This is independent of the QIAstat panel. It's really more in relation to the absolute value of a syndromic approach within the context of this pandemic and the potential overlap of this pandemic with the upcoming flu season. I think that syndromic solution is really very closely linked, first of all, to rapid turnaround time. It would be non-actionable to provide results on over 20 targets without the ability of taking remedy and taking a patient management decision within a very short time. And this COVID-19 pandemic is really highlighting the importance of time to diagnose. Rapid diagnostic enables faster response, even in the absence of treatment. And we knew from the beginning that key to curb this epidemic was and still is time to isolation and diminishing transmission in the community. These are just two examples from the literature depicting this. To the left is an older study by Fraser and colleagues, which found that pathogen transmission rate to be influenced by duration of contagiousness of the individual. And again, very much that related to time to isolation and therefore determined by time to diagnosis and identification of infectious sample. But to the right, even more relevant, is a study from Helwell. It's actually a model where they utilize a model assessing isolation and contact tracing and their ability to control transmission of COVID-19. What is very clear from this model is that the delay between symptom onset or diagnosis and isolation had the single largest role in determining whether an outbreak was controllable, particularly when the R0, the basic reproduction number, is between 2.5 to 3.5. So again, even in the absence of a vaccine or of a treatment intervention, the ability to quickly diagnose and quickly differentially diagnose, and therefore the ability to isolate the patient and to implement contact tracing is key in controlling the pandemic. But let me go now into 3 clear examples of why syndromic approach is relevant. The first example is from the pre-pandemic. These are 2 sets of data, again from Paris Hospital and from surveillance data from the Marseille Institute. To the left is a collection of data from the flu season and presented cases during the flu season from 2011 to 2016, demonstrating the heterogeneity, even during the flu season, of the etiological agent behind the influenza-like illness presenting at emergency departments. And you can see here that knowing whether it's flu is probably not enough in a proper differential diagnosis. And you can see here across November, December, actually flu represented but a very small proportion of cases. But even in the few weeks preceding the appearance of SARS-CoV-2 in France and in the region that were participating to this surveillance exercise by the Marseille Institute, was very clear that there was plenty of culprit for respiratory syndrome, but not SARS-CoV-2. So this became really crucial in the early days when SARS-CoV-2 was not yet prevalent, and the positive predictive value of symptom presentation was very, very low. The ability to quickly distinguish, even in the absence of a SARS-CoV-2 test, to distinguish the etiology behind the respiratory presentation was crucial. But a very clear example came very early on from the National Institute for Infectious Disease in Italy at the Spallanzani Hospital, where they utilized the previous generation of the respiratory QIAstat respiratory panel, the one with the 21 respiratory pathogen except the SARS-CoV-2, so the old generation, which they run in parallel with the WHO protocol for SARS-CoV-2. And what was very clear, this was in the early day where there were a handful, these I think were 3 cases that you can see here of SARS-CoV-2 that were detected. But you can see the importance within the emergent pandemic and being able to discern the viral cause or in few cases bacterial pathogen behind the presentation. It was crucial and remains crucial. And you can see here that up to 40% of cases were actually distributed among more than 10 different pathogens. And this proved to be crucial in the early days when integrated syndromic panel with SARS-CoV-2 were not yet in existence. And I like really to quote the conclusion from the Spallanzani Institute, where they highlighted that in an epidemiological situation with low circulation of SARS-CoV-2, and let's remember, this is not a thing of the past, because we could, and we are re-encountering this, for example, in Italy, in certain regions where the circulation is close to nil, this ability where alternative diagnosis may clarify an individual patient risk, allow adjusting public health containment measure. And I think this is crucial because this will be a returning rationale. What we are learning the hard way is that this pandemic is likely to have waves in which we will have isolated outbreaks embedded in area of low endemicity. So this ability of very quickly discerning among respiratory pathogen will be critical in adjusting the public health response. And lastly here, I have 2 examples coming instead from the peak of the epidemic. Now, I don't want to go too much into the details because there's now a very high volume of paper being published on co-infection or alternative presentation to SARS-CoV-2 with overlapping symptomatology with other infection. The key here is that there is evidence, and increasing evidence, I would dare saying, of a heterogeneous and unpredictable picture of co-infection, which makes the case for a syndromic approach within the pandemic even stronger. What I'm saying here is that it's going to be very difficult to identify a combination of 3 or 4 targets which would define 90% of the ILI presenting in the upcoming winter flu season. Because what is being demonstrated here, and I have two examples here, we can be in a situation where 20% of viral co-infection, where we have an additional 7% of bacterial co-infection, as well as situation in where the co-infection is mainly driven by isolated outbreaks, and we see very frequently now, particularly in nosocomial as well as nursing home outbreaks, there is a huge overlap between mycoplasma, for example, pneumonia outbreaks, we've seen a couple reported to us, and SARS CoV2 presentation. So the ability to be able to address this unpredictable heterogeneity in terms of concomitant epidemiological presentation, we feel will be crucial, especially in those settings which will need to adapt very acutely to the re-emerging flu season, where you will have a disparity in presentation and etiology behind the influenza-like illness present. And lastly, I just wanted to spend a word on city value. Here are two examples on how city value is becoming of importance in assessing potential clinical management. You can see on the left the correlation between city value and severity of cases, where city value, which are inversely correlated to viral load, remains significantly lower for the first 12 days in more severe cases comparing milder cases. And to the right, a similar example where the viral load in MPS sample was highest during the first week of acute symptom presentation. Actually, the evidence around city value in COVID-19 has exploded in the sense that what is really noticeable is a very close correlation between city value and clinical prognosis and evolution. We have actually just concluded a systematic review, which we have now submitted, and we hope it'll be publicly available soon. And lastly, I would just like to conclude on what are we expecting. I gave you some very personal epidemiological thinking in terms of what the control is going to be like and the requirement for testing, as particularly in the Northern Hemisphere, the flu season will be approaching, and where we get also into a outbreak-driven epidemiological situation. But I think the big question mark, given the known behavior of the existing coronaviruses, is whether COVID-19 will establish its seasonality. If that is the case, that will be one more element adding to the syndromic potential of ILI presentation and more etiology overlapping around the acute presentation of upper and lower respiratory syndrome. So hopefully the advent of a vaccine will be able to modulate and moderate that seasonality, but again, it's something we need to take into account, and we feel probably within that context of seasonality, there is a clear role for syndromic testing and providing a quick ability to actionable results around a number of pathogens, covering for the majority of possibility in terms of respiratory etiology. And with that, I would like to conclude and open the floor for questions. Thank you very much.
Thank you to Dr. Davide for giving us such an excellent speech. It's very clear and precise. And I think the panel is of great help to us when winter comes, that we are having all kind of virus, and especially influenza, and also the COVID-19 will appear, or even now we have flattened the curve a little bit. So I would like to see any questions from the audience. Okay. Let me ask one question to Dr. Davide first. The thing is that now the panel is distributed all in the Europe and also the United States. So in the Asia countries, so now we already have the panel or not?
So if I understand the question well, you're asking concerning distribution in the Asian market? Yes, indeed, it is available, and my commercial colleagues would be able to give you more indication on where it is registered within Asia. But yes, the distribution is worldwide. Obviously, there might be local differences in terms of local registration, which might delay the availability in a single country, but it is available worldwide for distribution.
Now there's another question coming out from the line. Dr. Davide, could you please elaborate why QIAstat diagnosis panel get rid of gene detection of SARS CoV2? Is this related to viral genomes replication? I mean, once we detect RD, PP, and E target, is enough to confirm SARS CoV2 exists? This is the question.
Yeah, thank you. That's a very clear question and a common one. So let me just take you back to my earlier comments during the recording on how we came to the conclusion that the combination of the E gene and the RDPR region combination was the ideal one. So it was done on the basis of maximizing the analytical sensitivity and specificity in the cartridge, so in the specific solution that we have. We tested the various approaches in terms of combination of genes and selection of genes, including the N gene, and the combination that we've chosen is the one that for our particular platform and cartridge setup is the most effective and efficient from a sensitivity and specificity point of view. Now, to further corroborate that point, I was pointing out at a publication that we have just had approved in the Journal of Infectious Diseases and Therapy, highlighting the maintenance of sensitivity against the variants that are circulating as of week 21, to further corroborate and strengthen the point that the choice of the E gene and the RDPR region were indeed so far proven to be efficient particularly from a sensitivity point of view.
Okay, I think there's no more questions from now. So we thank Dr. Davide for your excellent speech, and thank you for your attending.
Thank you very much, Dr. Wang, and it was a pleasure. Thank you very much.